Okay. All right. Okay. One more. Okay. All right. Admit one more people. Then let's start. Okay. All right. Okay. All right. Okay. Just about to start. People keep joining. Okay. All right. Last person. Okay. Good morning, everyone. Or good morning in Toronto, afternoon, evening from wherever you're signing in from. And so, as I admit people here, I'm just going to start. So welcome to our first KCN event, that is Crumble Computational Neuroscience event for 2021. And so for those who don't know, let me just give a little bit of a background in terms of these KCN events. So we started them back in 2017, and the goal is really about bringing together researchers and trainees at the Crumble and the wider community to really sort of talk about mathematical, computational, theoretical, neuroscience type issues. And we encourage it to be sort of interactive, but of course we're somewhat limited with Zoom, being virtual. But really, it's about trying to raise awareness of different issues in the field and catalyze the development of new collaborations, new research directions, learning from each other. And so today we're very happy to welcome Dr. Travis Craddock. And Travis is a professor, an associate professor in the departments of psychology, neuroscience, computer science, clinical immunology at the Nova Southeastern University in Florida. And there he serves as the director of the clinical systems biology group at the Nova Institute for Neuroimmune Medicine, where he's applying computational systems biology and biophysics methods. And so, there's more on him online, but his areas of focus are cellular information processing, molecular neuroscience, biophysics of neurological and neurodegenerative diseases, systems neurobiology, quantum biology. And today I'm very pleased to welcome him. I should tell you a little bit about him. He has a PhD in biophysics from the University of Alberta, where he focused on neurodegenerative disorders. And today he's going to talk to us about light-based quantum processes in the brain. And so just to let you know that we welcome sort of questions. Travis has no problem answering questions in the interim. I will keep an eye on the chat if somebody has something there or if they raise their hand, but also he'll be around for questions after. So without any further ado, welcome Travis or Professor Craddock, and take it away.
Thank you very much, Francis. Yes, thank you for inviting me here. I wanted to talk to you today about potential for light-based quantum processes in the brain. That's why I have the question mark there at the end of the title. Francis said that these kind of events were meant to generate discussion, so I'm hoping that that I can show you some interesting information and hopefully generate some thoughtful discussion on this area that I think is important. Let's see here. First, I want to thank Crumble for inviting me to give this presentation. I really appreciate it. It's been a while since I've talked to people up in Canada, being stuck down here in the US, especially with COVID going on at this time. As Francis said, I work at Nova Southeastern University. We're based in Fort Lauderdale, Florida, where I work at the Institute for Neuroimmune Medicine. Our main goal is to find new diagnostics and treatments for illnesses involving the neuroinflammatory process. So we involve four main cores. We have a functional clinic, we have a biological laboratory, a wet lab, an animal core, and then I head up the clinical systems biology group. So we do all of the modeling and data analysis for the team. This is my current team. We've been going through a lot of flux during this pandemic time. So we've had certain people drop out and new people join the team. But we're starting to pick back up again now that everyone's getting used to this kind of online environment. Now, the work that I'm going to be talking about today is, again, primarily theoretical. Most of the funding has come from seed grants through Nova Southeastern University, as well as through the Army Research Office, where I've been working with a collaborator at City College of New York, looking at light interactions in the brain. All of the kind of simulations that I'm going to be talking about were all performed through a collaboration down at the University of Miami, where we use their high performance computing platform Pegasus to run all of our simulations. This is a general outline that I'm going to be going through. So one, I'm going to basically go over the clinical diseases that may be of interest, that are of interest to me, but have kind of bearing on the kind of things I'm going to be talking about in regards to light in the brain. Then I'm going to go over a brief summary of what ultra-weak photon emission is, which is what light in biological systems is referred to as. Its potential for diagnostics, so new diagnostic avenues, the potential for functional roles. So not only is this something that is happening in biology, but it might actually be used by biology for signaling within the brain. And then I'll talk more specifically about some of the light matter interactions that I've been modeling with how light interacts with the neuronal cytoskeleton, and then hopefully conclude everything. As Francis said, as we go through this, if you have questions, feel free to ask them. I'm not sure. I think Francis is going to be monitoring for when they pop up, because I'm not quite used to doing that through Zoom at this point. So we'll see how that goes. Otherwise, I'll be able to answer them at the end of the talk. Okay, so the illnesses that I'm primarily focused on, especially in regards to neurodegeneration, are tauopathies. So you may be familiar with Alzheimer's disease, which I think is the most prevalent and well-known tauopathic disease. Other related diseases are things like chronic traumatic encephalopathy, which has an AD-like pathology, but takes place within the cortex as opposed to the hippocampus, which is seen in Alzheimer's disease, as well as Parkinsonism in several other illnesses. So all of these share a common dysfunction in the protein tau, and by extension, the microtubule cytoskeleton. Broadly, tauopathy is a neurodegenerative disease. It is the progressive loss of neuronal mass and function within the brain over time. Your neurons effectively wither away and die as the disease progresses. And you can see this here comparing normal and healthy brains with both Alzheimer's disease and advanced CTE. Now, what drives this kind of degeneration is the hallmark of these kind of degeneration for tauopathies, at least, are the presence of neurofibrillary tangles. So this is a staining, again, in control versus CTE and Alzheimer's disease, showing these dark stains for tau, or a stain for what's known as a neurofibrillary tangle. And what that is if you have a tau protein, which is a long, flexible protein, through various modifications, truncations, mutations, hyperphosphorylations, these things can start to stick together into oligomers and eventually into filaments that clump together into what is known as a neurofibrillary tangle. So these are misfolded proteins that are sticking together and forming aggregates within the brain. It is a hallmark of the neurodegeneration. Where it's coming from or where it is within the stream of the neurodegenerative process is not known. It's not known whether it's a cause of the neurodegeneration or if it's a result of the process that gives rise to the pathology. But it is something that is seen within all of these these tauopathic neurodegenerative diseases. Now, getting closer, a neurofibrillary tangle, this protein tau, its main role is to stabilize microtubules within the brain. So microtubules are long, hollow tubes that form the architecture of the neuron. So they're responsible for the neuronal processes of dendrites and axons. They couple together through cross-linking proteins. Now, if you're looking at these things in cell culture or sort of normal cell types, or just within assays, they tend to go through a process of growing and shrinking known as catastrophe and rescue. Now, it's unique within the brain. In order to stabilize them, they have these microtubule associated proteins, tau being one of them, and they decorate the surface of the microtubule, and they serve to stabilize it so that it remains stable in the protein, within the neuron, to give it that the characteristic shape that you see with the arbiter of the dendrites and the axon. Now, in these tauopathic diseases, whatever the dysfunction is in tau, whether it's hyperphosphorylation or mutation or something else, it prevents them from sticking to the microtubule. So they don't bind to the microtubule, and therefore the microtubule becomes weak and it starts to depolymerize through its natural processes. And when that happens, you no longer have the skeleton that holds up the axon, and therefore the axon starts to wither away. You start to lose synaptic contact, and you're basically start to generate your neuron as it withers away and dies. And the result is these clumps of the neurofibrillary tangles. Now, the things that generally drive this are, either direct events, upregulation or aberrant activation of the tau kinases, mutations, covalent modifications of tau, as well as indirect events, toxicity mediated by other misfolded proteins. And then the two that I kind of focus on are oxidative stress and inflammation, and those two kind of go hand in hand. And what I'm going to be talking about the rest of the talk is going to really focus on oxidative stress. Now, I'm going to change gears here for a second as we go through the rest of the material, but I just want you to keep in mind this idea of tauopathic illnesses and the role of oxidative stress as we go further here. Okay, so switching gears here to ultra-weak photon emission. So this started out as a very, very controversial field, but it's been shown time and time again that all living tissue produces a weak radiance. So they're constantly emitting photons. Now, this was originally discovered in Russia by Alexander Gurwitch in 1932, where he was looking at emissions from the onion plant. And since then, it's been shown in every type of living system. So bacteria, plants, mammals, any type of cell, that you can measure this very, very weak photon emission. And the wavelength of these photons as they're being emitted from the cells range across various frequencies. So they can range from the UV through the visible and into the IR frequencies. And it's believed that they have different sources depending on where they're coming from or what they interact with along the way. Like I said, it's very, very weak, ultra weak. So the intensity is 10 to the negative 16 watts per centimeter squared. And that's 3 to 6 orders of magnitude lower than what's visible to the naked eye. So it's very, very weak. It's not something that you could ever see directly. But with sophisticated cameras that take binning snapshots of living tissue within a dark chamber, eventually you can build up a profile of the light that's being emitted, and that's what's shown in the lower right-hand corner there. So we have figure A is the photograph of the hand, and then over time you can see the resolution of the biophotons, as they're called sometimes, starts to pick out the shape of the hand as more and more time passes. You can see the shape of the hand start to form. It's referred to as ultra weak because of the intensity. It's also been referred to as biological autoluminescence, and colloquially they're referred to as biophotons. However, that term biophoton has tended to be co-opted by New Age literature, and therefore it's not favored so well in the scientific literature. So I'm going to stick with referring to it as ultra weak photon emission, or UPE, for the rest of the talk as much as possible. Now, where does this come from? So for years they've been looking at how does this ultra weak photon emission occur, and what research has shown is that it's related to oxidative metabolism. And so during the oxidative metabolism process, as you generate ATP, you tend to generate reactive oxygen species. And as these reactive oxygen species recombine and decompose, and the system clears out the reactive oxygen species, what you can get left with is electronically excited species. So you can get carbonyl groups that are in a triplet excited state, or a singlet oxygen, which is in a singlet excited state. And when those relax from the excited state back down to ground state, they release a photon of energy. And that photon of energy can either be emitted into the system, and then it's free to interact with whatever is surrounding, or it can be transferred through non-radiative processes to other molecules that may be excited at the same or similar wavelengths. Now, as you can imagine, because we're talking about neurodegenerative diseases, and in neurodegeneration, you tend to have an increase in oxidative stress. you have a higher amount of reactive oxygen species, you can imagine that you will have more of these biophotons being emitted. So in neurodegeneration, you have these elevated ROS levels, which are very, very toxic to the cell. They damage DNA, RNA, proteins, membranes, lipids. Everything within the cell is kind of game for ROS to interact with. And it leads to damage. So this is seen in aging and the pathophysiology of many diseases, in particular neurodegenerative disorders. But how that damage is incurred is not really known. So what is the physical mechanism by how the damage is mediated? Is it through the scavenging of electrons? Yes, in some way, shape, and form. But it can also be due to the presence of these high energy photons, which can then interact with sensitive molecules and potentially cause damage. So that's kind of where these things start to overlap and interact.
Travis, just to interrupt you for 1 second, there's a question in the audience. Jeremy, you want to unmute and ask?
Yeah, hi. Hi, Travis. So I have a question that might be very, very simple, but most living organisms, they have heat, right? They generate heat through the various metabolic processes, and some fruits are radioactive as well. Like bananas emit gamma waves. So I'm wondering, like, is it so surprising that biological tissue emits light in that regard? So they radiate.
No, it's not. It's not surprising. It's just that these whether these photons are, or this light that's being emitted, whether it has it is just waste energy, or is it serving a purpose within the cell? That's the question of the day, really. Is it useful energy? Is it being used by the system for something purposeful, or is it just being let off as heat to the environment? In regards to is it controversial? I would say not now. I mean, there's plenty of things that have shown that the biology lets these off. But back when it was first discovered, the initial works of Gershwitz and whatnot were quite controversial, especially the photons that they measured in the closer to the UV spectrum is something that is kind of surprising.
And there's a follow-up question after Jeremy. Sorry, Jeremy, were you done?
Yes, I was. Thank you.
Okay, all right. Andre, do you want to unmute and go ahead and ask?
Yes, thank you. Hi, Travis. I just wondered to follow up on Jeremy's question, if it's known what the spectrum of this radiation is, and if it relates to just normal black body radiation.
No, yeah, it doesn't relate to normal black body radiation. There are spectrums that are measured, and you get different spectrum depending on the source that you're looking at. So there's a recent paper, I'm not presenting it here, but a recent paper, I think it came out either this year or last year, showing that different species of bacteria emit different spectral profiles of light. So it is somehow tied to the system of interest and what is going on, or what process it is undergoing at the time.
And the intensity is lower than black body, I guess.
Yeah, it's extremely weak. Yeah, definitely.
Okay, thank you.
Okay. So because of this, because it's tied to this metabolic process, especially oxidative stress, it's been more and more it's been looked at to be a potential non-invasive health assessment mechanism. So you can imagine, a lot of diagnostics that are run in health involve, you have to go into the system and manipulate it in some way. Either you have to draw blood, or you may have to give a radioactive marker, or you somehow have to perturb the system. And in this way, because you're just measuring the natural radiation that's coming off the system, it may be a way that you can non-invasively assess what is going on underlying given dysfunctions. So it's been shown that this photon emission can be used as a dynamic tool for monitoring oxidative stress. It correlates to inflammatory metabolism and oxidative stress in inflammation. In this case, it's arthritis. And it's also correlated with cerebral energy metabolism and oxidative stress. So these are just a few of the papers that are out there. So it does look like it is a good correlation between oxidative metabolism and reactive oxygen species and what is actually being emitted. This is an example of, this is from quite a while ago, but it's looking at a mouse model of arthritis. And on the far left there, you have the animal. That's the photograph of the animal and the given limbs that they're going to be looking at. And then the top row is looking at the biophoton profile coming off the animals. The middle column there is in the absence of any additional treatments, and on the end they add a molecule called luminol to enhance the emission. So the bottom row is the rheumatoid arthritic animal. The top row is the control, and so especially in that middle panel, you can see that you're getting quite more biophoton emission from the limbs in the ill animal. Now it's also, there's possibles for using this as diagnostics in the brain. This is again quite some old work, but it's looking at the correlation between photon emission and the theta wave component I believe it's in a rat brain, a rat cortex. So the bottom panel is showing a very poor resolution of the animal's brain and the biophoton profile, the image, and then they put it next to the theta wave component that they measured through EEG, and it shows a fairly good correlation between it.
Sorry, Travis, one question from Jeremy again.
Mm-hmm.
Sorry, I won't do that all the talk, I promise. I'm just wondering, is there a link with this sort of bioluminescence, I don't know, and inflammation? You would suspect that some spatially clustered.
Yeah. So yeah, I want to make a distinction here between the two here. So you have the process of bioluminescence, which is usually generated through chemical means. So you can think of like a firefly and whatnot, or glowworm as it illuminates. So that would be bioluminescence, and this is a completely different process. So in the field, they tend to draw a distinction between the two. So this is autoluminescence being generated by the metabolic process on its own, not a specific chemical mechanism. I mean, obviously it's through the ROS, but not through a chemical mechanism which is designed to generate the light for us.
Yeah, okay, but just in terms of inflammation, because I suspect that some more active sites, that are ongoing, some major structural or metabolic changes could express more of that.
Yeah, so I mean, if you ramp up your metabolism as you're generating, more ATP, you're going to be generating more ROS, and therefore you're going to be generating more biophotons. So definitely in inflammatory processes or highly active brain areas are another areas that they would expect to have higher photon emission. But yeah, definitely within the inflammatory process or any overactive metabolic area is where you'd expect to find a greater intensity.
Okay, thank you.
So this same group here, I mean, the correlation I'm not particularly happy with in this, but there is a trend there. So on the left here, you have the biophoton count versus the theta wave component, and then that's where they put a window in the skull. So they remove the bilateral bones and measure through a window into the brain. And then on the right, they actually measured without removing the bone. So it's measured through the skull, and it's less, but there's still kind of that trend that's going on. And so the idea is that this may be potentially something that can be done non-invasively through the scalp. This is some other work. This is with work from my collaborator, Dr. Alfano, down at City College of New York. And what they've been using terahertz spectroscopy of brain tissue in Alzheimer's disease and Parkinson's. So what they do is they send laser light through brain tissue and then measure to see if there's a difference between healthy tissue and ill tissue. And what they find is that they get these differences in peaks, peak shifts between the Alzheimer's model and the control model. Now this is different. This is not biophoton emission, but it is putting light into the brain. But the reason I'm bringing this up is that you could expect, I mean, if you're adding light to the system and you're reading out a different signature, that if there is endogenously generated light within the system that's already coming out, and you have a typically healthy system versus an ill system, you can expect that you'd probably have a different profile of the emissions, and it may be due to what's going on underneath here. Here they're talking about free tryptophan, so that there's more free tryptophan in the AD tissue than in the normal tissue. But you can imagine that you may be able to pick this up if there's some sort of emission coming from the brain. So the idea then here would be to look for those peak shifts in ill tissue, and then that would be your diagnostic for measuring, you know, what is going on underneath the scalp. So potentially as a advanced diagnostic to, you know, spot the initial stages of the illness. Again, this is all theoretical. It's just kind of ideas that we're batting around. Okay, so yeah, there's a potential for diagnostics here. We could potentially measure this to see what is going on with the stress that's underlying the system. But is this just waste energy? Like, is it just something that happens to arise because of the, you know, the chemistry of reactive oxygen species metabolism? Is it used by, did biology evolve to take advantage of this high energy? I mean, there's, you know, if you're ranging from UV into the visible and then down further into the IR, I mean, that is a lot of energy being generated by the system. Is it just being let off into the environment for, you know, as waste, or did biology evolve to take advantage of the very high energy that it's generating? So this paper here, I was looking at activity-dependent neural tissue oxidation emits these photons. So I mean, that's no surprise. If you have more active tissue, you're going to be, you know, ramp up your metabolism, generate more ATP, generate more ROS, and you have more photons that are coming out. But it's also tied to electrochemical signaling. So this is the group in, I believe it's in Japan, where they're looking at if you introduce glutamate into the brain, how does it change the biophoton profile? And so glutamate, because it's, you know, one of the, you know, most prevalent signaling molecules in the brain, that's why they looked at it. And they found that as you introduce glutamate into the system, the relative emission profile increases and remains stable. So that's what's shown here. So this is a photograph of the brain itself. The gray value and the biophoton number, they're, you know, analogous measures. They're slightly different in the way that they're calculated, but you can see that the general shape and profile is the exact same. The top panel here, D, is the relative gray value, and E is the actual biophoton count. And so what they're doing here at the initial, that's this initial arrow here, is they're introducing, I think it's 50 milligrams of glutamate within the brain, and then they measure how it changes that biophoton emission, and it ramps up over about 90 minutes or so, and then it remains fairly stable for a long period after that. So you get, you know, a large outflux of photons compared to what it would be without the glutamate in the system. What they do here then, where you get this weird stuff going on, is they take out the slice. I believe these are brain slices. They wash it out and then reintroduce glutamate, and they get this more exacerbated response that is quite pronounced. I'm not so much interested in this. I feel that they're manipulating the system too much, but still, this portion down here below 300, you can see that at least this signaling molecule can lead to greater emissions, and so somehow the metabolism is related to the electrochemical signal, and therefore it ties in with the emission that is being released. And then this one is the one that I really like. So this one came out this year, or sorry, last year, which we had New Year's. So in the brain, deep within the brain, there are light sensitive molecules. There's opsins. So in your eye, you have opsins within your eye that pick up light particles as they come in, and then these are light sensitive molecules that change shape, and then in that change of shape, they set up protein conformational change, which eventually results in electrical signals that get picked up by the brain, which gets interpreted as vision. But in the deep brain, there are also opsins known as opsin 5 or neuropsin. For example, it's found in the hypothalamic preoptic area. And these are sensitive to light in the UV region. They don't know exactly what it is, but this work here, they were illuminating it directly. So they put a little probe into the brain, and they went into this area, the preoptic area, and they stimulated it with violet light and ultraviolet light. And what it was found was is that it stimulated the neuron, and then it that leads to the suppression of brown adipose tissue thermogenesis. So this light that they introduced directly into the system can trigger this effect to affect this thermogenesis within the animal. So then, if you have this system in there, which is generally sensitive to violet and ultraviolet light, why is it there? It's potential that the functional role could be tied to this UPE. So if you have UPE that is letting off this endogenous violet and ultraviolet light, these opsins could be picking it up and could be somehow changing your thermogenesis within the system. And if this is one system that is could be influenced by this UPE, then there may be other systems that we're not aware of yet. So I think this is quite interesting, this type of work. Was there a question? I just saw a name pop up on my screen.
No, I don't see any questions. Yeah.
Okay, so now if we go into some more theoretical work, this is work done by Christoph Simon out in Calgary. So one, we're talking about, you had this potential for diagnostics by this endogenous light that's being emitted. There's a potential for functional role by it triggering light sensitive receptors within the brain. Now this starts to get into more physical ways of viewing the light. So it's considering, if you look at the axon and consider it to be a fiber optic cable. So a myelinated axon, your index of refraction within the axon is going to be different than the index of refraction of the myelinated sheath. And because of the way it's set up, you have a lower central index of refraction compared to the outer core. And this is exactly the setup of a fiber optic cable. And what that allows is that if you have a light that's traveling down this system, it can bounce down it can refract within the core, and potentially transmit a photon from one end of the cable down to the other. So the idea is considering the myelinated axon as this kind of cable. So what they did is they set up a model. They started with a simple model of just a circular axon or cylindrical axon with a cylindrical myelin sheath, and then they started to add in more details. So they added in nodes of Ranvier. They added in different shapes for the myelinated sheath, so it wasn't a uniform cylinder on the surface of the axon. And then they changed the shape of the axon itself. And what they did was they solved Maxwell's equations for the eigenmodes of how electromagnetic radiation could propagate along this cable. And what they found is that depending on the parameters, so depending on what the wavelength of light that was used and how much deformation that you had, that you could get a fair amount of transmission within the system. So over there in the bottom panel C, you can see that for small changes in the inhomogeneity of the axon, and depending on the wavelength, you can get somewhere near 100% transmission within the system. And so they came to the conclusion that myelinated axons can serve as a photonic waveguide. Now, again, they did add in imperfections within the axon itself. However, they didn't take in full details of what is actually inside that inner core of the axon. So, instead of considering it as a homogeneous medium with a few imperfections, really, when you look at an axon, it's filled with plenty of material. You have lots of protein and whatnot in there. I'll get to that in a second, but here we have, this is actual in vitro proof that axons may actually act in this manner. So this group, headed by Sun, what they did is they came up with this novel setup here. So you have this incubator, this illumination plate which has holes in it, and it's illuminated by various colored LEDs at the bottom. you have this incubation plate, which is shown over here. So they have a hole which lines up with the hole in the illumination plate, and then the channel, which the nerve root is sent through over to a well where the cell or where the silver, I think it's silver nitrate is located. And silver nitrate, when it's exposed to light, will darken. And so you have this, the only way that the light can get from, hole A, where it's coming in from the LED, over to a hole B where this reactive substance is by passing through the channel where the nerve root is. And so they illuminate it with different wavelengths of light, and they found a significant increase in all the wavelengths, from infrared all the way down to blue and white compared to control. So the control is having no light illumination, and that was both for motor nerve root and sensory nerve roots. And then they went further, and they added in 2-deoxy-D-glucose and sodium azide, which is a metabolic inhibitor, and as well as they added in procaine, which is an anesthetic, and they found that it reduced that transfer of the biophotons from one end of the nerve to the other end of the nerve. So it seems to give evidence that the nerve root can act as an optical channel to transmit light from one end to the other, which I think is really cool. I really think this is a great setup. I think it's a great experiment. Now, where I come in, where I'm interested is in that axon. As I said, it's not really a homogeneous core with some minor defects, but it's filled with other proteins, but particularly it's filled with a lot of microtubules and mitochondria. So they're packed in there. Now it's known that microtubules in neurons, they co-localize with mitochondria to a high degree. So wherever you're finding mitochondria, they're always surrounded by microtubules. And that's because microtubules anchor them and are used to transport the mitochondria around the system. And likewise, mitochondria are letting off energy molecules like GTP that are responsible for, that microtubules need to polymerize properly. It's been shown that microtubules themselves, so what I'm trying to say with that is, because they're so close to one another, if you're getting a lot of this biophoton emission coming directly from reactive oxygen species, and it's coming from oxidative metabolism, the mitochondria is going to be the prime source of that. And because microtubules are so close to it, they're bound to be one of the first lines of defense to pick it up. It's been shown that microtubule polymerization is sensitive to photostimulation in a dose-dependent manner. As well, like Christoph Simon's work, there's been other work by Pokorny in Czech Republic, Sifra and Tazinski, that show that microtubules themselves can act like optical waveguides. So they can function almost analogously to the axon, because they have a central core which has a different index of refraction compared to the protein outer layer itself. So just going a little bit further, microtubules, they form this elaborate architecture within the neuron. They bundle together with things like tau, which hold them together within the axon, or MAP2, which holds them together in the dendrites. And they provide the overall structural support for the neuron. And almost in every single cognitive disease in one way, shape, or form, there's some sort of misregulation or dysregulation of the neuronal cytoskeleton. So in Alzheimer's, in the Tauopathies, but also in things like bipolar disorder and schizophrenia as well. I think even in Down syndrome and Fragile X syndrome, there's microtubule defects that have been shown. Microtubules themselves are formed up of a protein called tubulin. It's a heterodimer made up of alpha tubulin and beta tubulin. And these things are naturally sticky, and they self-assemble first into long filaments called protofilaments. And then these protofilaments stick side to side, and then they wrap around to form the helical microtubule cylinder. So there's a hollow core in the center of it with this protein wall on the outside. They're responsible for things like I said, maintaining the architecture of the axons and dendrites, but they're also the train tracks that motor proteins walk up and down to carry around cell cargo. So they're quite important to overall neuron function. Now, coming back to this this idea of how can this photon emission affect proteins. One is just with interaction of light. So if you have a photon which is high enough energy and it interacts with the protein itself, it can cause damage. But this is the mechanism here that I want to focus on. So as you go through here, these photons here, these are emitted photons. So these emitted photons are radiated by the system. So as the system is excited, it's excited at one wavelength and then it emits the photon at another wavelength. And it's usually a down conversion. So it has less energy because the protein uses some of that energy and it's setting up vibrations and whatnot in the system when it gets excited. Now, this process here to go from this excited triplet carbonyl to an excited chromophore, this is a non-radiative process. So if you're familiar with the process of FRET or resonance energy transfer, you can have energy transfer between two similar chromophores as long as they excite at the same wavelength. So there's there's a coupling between the dipole set up in the system, and the energy can be transferred from one to the other through this non-radiated process. So what I'm particularly interested in is the high energy end of those biophotons. So yes, you can have visible light, your yellows, greens, and blues being absorbed by chromophores within the system, like flavins or heme groups can also absorb. So they can become excited by these photons. But those high energy photons, which they're not observed a lot within the system, so they're very, very conserved. They have been observed, but they have been shown to be created. But they're also picked up by one of the most prevalent chromophores within the cellular system, which would be the aromatic amino acids. So there's way more aromatic amino acids within the cell structure than things like flavins or heme groups. Because of that, these UV photons that are being emitted are more likely to be absorbed and then down converted to a lower energy before being emitted than some of these other photons, which may not have readily available chromophores to absorb them. Now, what happens when tryptophan, in particular, gets excited by a photon? There's a reconfiguration of the electrons within the system as you go from the ground state to the excited state. And as those electrons shift between the two configurations, it sets up what's known as a transition dipole. And these transition dipoles are due to the oscillation of the electrons going from the ground to the excited state. Now, in doing so, this transition can then be shared across multiple chromophores that have a similar excitation energy. So tubulin itself has 8 tryptophan molecules within it, and set up in this configuration. And at the time when I started doing this research, there was work looking at the photosynthetic complex, and the idea was that the biology was way too warm, wet, and noisy to support any sort of quantum process. And then in 2007, Greg Engel's group had some measurements that suggested that there was actually quantum effects that were going on, and it was due to delocalization of photon energy across a chromophore network of chlorophyll molecules. So at the time, I was doing modeling for neurodegenerative diseases and cancer, looking at tubulin, but I really liked this idea of this quantum process going on in biology, and I said, is there any way that this could take place in something like tubulin? So I looked at, this literature came up on biophotons in the UV region, started looking at tryptophan molecules because they're likely absorbers, and then looked at this architecture. And the spacing and energetics of this system are very reminiscent of the spacing and energetics that are available to the photosynthetic complexes that harvest and transfer light within plants. So we did some modeling following that the methods that they were using in photosynthesis. And I'll get to some of the results in a minute. But the idea is that you have these mitochondria, which are generating your reactive oxygen species. And as they go through that conversion process, they generate an electronically excited triplet carbonyl group, which can then transfer the energy within tubulin. So it can be absorbed. I mean, the carbonyl group could be in tubulin to begin with, and then that could set up a chain where it would excite a network of tryptophan molecules within the microtubule. It's lofty thinking, but it was a theoretical exercise to see if this is even possible. Like I said, we used this idea where we have this chromophore network, which is a setup of dipoles. So we set this up using a standard Hamiltonian, where you have creation and annihilation operators, which are generating site-specific excitations. So each one of these sites is one of the tryptophan molecules. And then you have coupling between the tryptophan molecules according to the point dipole approximation. We built models using molecular dynamics simulation. We simulated it to be at equilibrium temperature to get the atoms in plausible orientation. Then we calculated out what the excitation energy would be for the individual tryptophan molecules at each of their sites, because each one of them is going to be affected by its local protein environment. So we used density functional theory to calculate that out. And then, like I said, looked at how they couple to one another using this point dipole approximation to find out that this is how they kind of couple. So the strengths of the coupling between the tryptophans are shown here. So you have these kind of doublets that are very strongly coupled, and then weak coupling between those doublets. And then we use that to start to set up our model. We first used that coupling setup to calculate out the proposed absorption spectra of the tryptophan within tubulin, and showed that it aligned quite well with the absorption spectra, the circular dichroism, and the linear dichroism spectra of tubulin itself. And then we looked at how a single photon would perturb this system, and how the energy would be shared across the tubulin dimer. And we reproduced similar beating phenomenon that was shown by the Engel group within the photosynthetic system. We showed the same thing within the tubulin system. So this is something that potentially could be measured. The setup is there. You could use the exact same system that was used for the photosynthetic measurements. However, because it's much higher energy, there are a lot of technical hurdles that have not been overcome yet. I believe last time I talked to someone that was doing this, they said that they were 5 years away, and that was probably about 4 years ago. So fingers crossed to see what happens with that. However, because tubulin is not usually isolated alone, it's within these microtubule networks, there's the potential for this energy to be transmitted along the channel of the microtubule structure, because the tryptophans themselves form this continuous channel up and down the length of the microtubule. Oops. Where am I here? So we did further modeling with some collaborators down in Mexico and up at Howard University. And this was looking at how a single excitation can spread across the surface of a microtubule. And so what's shown here is if you excite just one single tryptophan molecule with the same sort of architecture that we had set up before, and give it some time, how far does it spread across the surface of the microtubule? And what we have here, this is the microtubule representation that's 100 dimers long. So it would probably be something like so 800 nanometers long is the length of this microtubule in this model. And what happens is that if you have a single excitation, depending on how you set the system up, within 2,000 picoseconds, this energy can be spread across the surface of the microtubule. Now, the rationale for this is that if these things can serve as an analogous type of optical channel, similar to what's going on with that was shown with the axon fibers, that these things can then funnel the energy not only just along the length of the axon, but can also funnel it to specific sites. So it might go to traveling, these things might be traveling to along microtubule associated proteins to tied in receptors. And if the receptors are light sensitive, then they might be able to trigger some sort of effect that might affect overall neuron function. That being said, this is all theoretical and hand wavy from this point on, but there are potentials here for this kind of light to have at least a transport role within microtubules, and then by extension within axons. Now, what you would expect in something like Alzheimer's disease or any other tauopathic disease where microtubules are falling apart is if you don't have this system to absorb that energy, you would expect that you would get perhaps you would get again an altered profile of the light that's being emitted, but you may be able to pick up more of that high energy photons because you don't have the system available to harvest that high energy light and down converted or transferred away within the system. So this might give an idea of what kind of profile you would want to look for. However, this is complicated by the fact that also in a neurodegenerative system, you have dysfunction within the mitochondria itself. And so how that would play in with what is naturally being emitted by the mitochondria versus what is being emitted in the neurodegenerative state is kind of an open question. But again, this is just for discussion.
Yeah. So, sorry, Travis, there's a question in the audience.
Yes.
Yeah, hello, can you hear me?
Yes, I can.
Yeah, so you're talking about excitation of tryptophan in photosynthesis?
This is in microtubules within brain cells or within any system. But the theoretical formalism that I used to set this up was based on the formalism that they used in the quantum description of photosynthesis. So that was just the impetus and the motivation to do this kind of study. But it's definitely within a mammalian system with microtubules.
I see. And when you talk about quantum formalism for photosynthesis, is that something other than the usual description of photosynthesis in the chloroplast?
It's the same system. So the classical description is that the chlorophyll absorbs the energy, and then that energy hops from site to site within the system and moves from the light harvesting antenna down to the reaction center where it's converted into chemical energy.
Right.
Now, every single description that they've used classically, I mean, you can describe it, but the efficiency that they measured in photosynthesis, you can't get it with a classical formalism. So they don't understand how they can get the efficiency that they're observing with their classical description. And that's because that this energy kind of hops from one site to the next as it goes. Now, the quantum formalism, what it does is instead of saying, the energy's hopping down the system, it actually gets spread out and is what's called delocalized across the whole antenna complex immediately upon excitation, and then eventually localizes back on the reaction center. And it's treating the excitation as a wave that propagates through the system instead of a particle that kind of hops through the system. Does that make sense?
Yeah. So
that's the difference.
Classical quantum idea of electrons are waves, and if we propagate as a wave.
In treating it as a propagating wave, you can actually get the efficiency that they observe. Whereas if you treat it as a particle, you don't get that. So that's why they appeal to the quantum description. I mean, there's a whole can of worms as to whether that's actually true or not, but it was the reason that I initially started looking at this. Okay.
Yep. Thank you.
So one of the places that this energy may be funneled to is places even within tubulin that may be able to absorb it at a slightly different energy and cause problems within tubulin itself. So for example, it's known that ROS can create or destroy disulfide bonds within tubulin, and it causes either depolymerization or aberrant functioning of tubulin. but the actual physical mechanism of how that takes place is not known. Now, disulfide bonds can absorb this same energy or the same wavelength of light as tryptophan, and can either create or break a disulfide bond. So this is just a potential mechanism that if the energy is transported to a site along the microtubule where it can then be absorbed by a disulfide bridge and then break it, then you can start to lead to the the photo damage that you would see within the microtubule that's known to be caused by ROS. So this is some of the work that regulation of the cytoskeletal dynamics by redox signaling and oxidative stress. They talk about this from a higher level. It's known that there is a physiological range of ROS that is needed for proper functioning, but if you have too much of it, then it leads to microtubule catastrophe. It starts to fall apart. In mammals and in the brain, it's unknown what happens if you have too little of that ROS, but in plants, it's been shown that low levels lead to aberrant formations of microtubules as well. So there's this like sweet Goldilocks zone of the kind of ROS that you need for the microtubule to function properly. And potentially these are kind of mechanisms that may be involved. So again, like I said, we're into the hand wavy portion of the talk.
So Travis, there's a question that,
yes.
Oh, yeah. Go ahead, Hattie.
Hi, I have a question about the tryptophan, interaction between ROS and tryptophan.
Mm-hmm.
Is there any possibilities, mechanism you have in mind for this interaction between these two?
Directly, you mean with the reactive oxygen species itself and tryptophan?
Right.
Yeah, so I mean, there are mechanisms that would be involved with electron scavenging, and I believe that tryptophan is able to not freely donate, but it has electrons that can be shared and taken away quite easily by anything that's reactive or a reactive oxygen group. But for the most part, in proteins, they are within the hydrophobic cavity of the protein, so they're not within the surface of the protein, so they're not readily available to be interacted with reactive oxygen species that are floating around within the cytosol. So there is a potential for that, but I'm not sure how that would work or how would actually become in contact so you can actually have that happen.
Right, because in NMDA receptor, there are aromatics like tryptophan, and there are also water molecules and this ROS. So I was just wondering, is there any kind of, this kind of situation for microtubulars?
Yeah, I mean, there could be. I mean, like I said, so I'm looking at tubulin itself because of its relation to these tauopathic illnesses. But you can consider that, I mean, almost every protein within a neuron is going to have its own tryptophan molecules that could be affected by this kind of energy. So for example, I know that there are serotonin receptors that on the intracellular side, so within the cell, they have a configuration of tryptophan molecules that, when irradiated with, I believe it's UV light, it can cause the receptor to open and close. So that, again, that's a potential way that this UPE can play a role. Not necessarily that it may, I mean, that's its prime role, that's what it's supposed to be doing, but even if it's a waste energy that the cell really doesn't want to use, if you have too much of it, then it may cause aberrant function within the system. So it may change the way that overall receptors are functioning within the cell.
Thanks. One last question. What's the lifetime of this excitation and for this network of tryptophan that they can?
The excitation, so the initial spread, so if you have like the initial excitation and then it spreads out, that takes place on the order of hundreds, maybe thousands of picoseconds, that initial spread where it spreads across everything. But the excitation of a tryptophan molecule, if you're talking about a singlet excited tryptophan molecule, it's on the order of nanoseconds.
Yeah, mostly I'm interested in this lifetime of this spread. that
Yeah,
when
so
it will
the spread is
die.
very fast. It spreads very, very fast, and then it tends to localize after the initial excitation. Yeah. So we're talking like yeah, definitely picoseconds, maybe up to a nanosecond.
Okay.
Yeah. I do want to say this as well, just because we're talking about this, is that tryptophan itself can also be can flip into a triplet state, and if it's excited in a triplet state, then the lifetime of that is much longer. So it's not on the order of nanoseconds, but it can last on the order of milliseconds to seconds or longer in this triplet state excitation. It's rare that it happens, but I mean that's we've been toying around with that idea that you could actually store some of this energy within the system in these triplet excited states to be pulled out later at some other time. But that's again, this is just playing around with some of the physics that's going on there. What the application of that is not quite known. But you can imagine here that if you have these systems, you can, and if this is a viable mechanism for how ROS damages the microtubule cytoskeleton, you can start to design treatments or chemicals or drugs that could bind to prevent or absorb the energy before it gets transferred into the system. So I know that there was some work using, I think it's methylene blue, where it generates, it's a singlet oxygen generator, and in doing that, it's been shown as a potential treatment for tauopathic illnesses, and it just seems reminiscent of this. Singlet oxygen ties in with this whole oxidative metabolism, and it may be a way that it's either adding more of these photons or absorbing more of these photons and changing the profile, which can aid or somehow provide a therapy for the system. So I mean, it's just different ways of thinking about how we could be coming about treatment avenues, as opposed to simply just binding a chemical and trying to change the protein conformation overall. Start to think about how these light interactions could take place, and how to change them might be a new avenue for treatments in the future. Okay, so in conclusion, the take-home message is that all living tissue emits this ultra-weak photon, these ultra-weak photons. It's tied to oxidative metabolism and the removal of degradation of reactive oxygen species. There's a potential for it to be used as a measure of oxidative stress within the system, and by proxy, then anything that would give rise to greater oxidative stress, such as ramped up metabolism or inflammation. The UPE can have a functional role in neurons through light-sensitive receptors, and it may serve as a communication channel, either via propagation down the axon as a waveguide or through microtubules as a waveguide. And understanding the role of this UPE in neurodegenerative illness could lead to light-based diagnostics for early detection or novel light-based therapies. And I think that's the end.
Okay, thank you very much. Okay, so there's some questions coming in, but I just want to take the opportunity to ask a very first question.
Yes.
Yeah, so as you were talking, I kept thinking optogenetics, right? So as obviously there's a lot of that going on these days. And so would that interact or how could you give me a sense of how much this?
Yeah, so the optogenetics is basically an engineered way of doing what I'm proposing here. So in optogenetics, if you don't know, is that you're introducing a light-sensitive molecule into the system, and then you're perturbing that light-sensitive molecule, which isn't normally there. You're perturbing it with an exogenous source to elicit a response.
Right.
And so yeah, I mean that's so the two things are different. Are one, that you're introducing the light-sensitive molecule because you want to control it, and two, you're using a more high-intensity exogenous source of light. Now, it's analogous to what I'm proposing here in that, you can see that using that kind of mechanism can generate a response. What I'm proposing here is that you would have endogenous light-sensitive molecules within the system, and then it may be taking advantage of this endogenous light. Now, because it's way weaker than the type of light that they're using in these experiments, The effects may be more contained. They're probably going to be more subtle if it's being used at all. If it's being used at all, that's the key. That's why there's a question mark at the beginning of this whole thing.
Yeah, no, I mean that's why. Like, so what I was wondering is like all these optogenetic experiments that have been done these days. Do you think they are the effect of the results of these experiments are affected by these?
Yeah, obviously. So for example, if they generate, it depends what wavelength of light obviously they're using. So if they use the wavelength of light that interacts with the light sensitive molecule that they've introduced, but it happens to also interact with some other light sensitive molecule within the system that can set up some sort of response, then yeah, it's gonna affect it. To what degree, I don't know.
Okay. All right. Okay. Thank you. So the first question here in terms of timing is from Alex. So Alex, I don't know if you want to ask it yourself.
Sure. So just a quick question. I'm curious, are light sensitive receptors present in human brains or just assumed to be based on rodent studies?
That is a very good question. I think they are in human brains, but I don't know off the top of my head. They're definitely obviously in rodent brains, but it may be assumed. I honestly don't know.
I'm also wondering if there's some non-invasive way of testing that in humans. Like
no. Well, I mean, post-mortem. You could look post-mortem.
Yeah, you could look post-mortem, that's for sure. I mean, probably also transcriptomic studies might give you some indication.
Yes. I'm pretty sure. I think it is in human brains, but I can't point to anything definitive, so I don't want to tell you yes when I'm not 100% sure.
Okay.
Sure. Thanks.
Okay. The next question is from Julio. Do you want to go ahead and ask it?
Yeah, sure. It's actually not Julio. I am
Maurizio.
Yeah, sorry, it's Maurizio. So hi, Travis.
Hello, how are you doing?
I'm doing fine, thanks. So I have a couple of questions, Travis. So the first one is a little bit technical. So I was wondering about this microtubule catastrophic breakdown. So it happens when you have excessive ROS production, right? So this excessive ROS production, it means that your tissue is already compromised. So this is like essentially the cell falls apart. So this is a very dramatic, extreme situations, right? So what is the clinical advantage of knowing this phenomena if actually when it happens, your tissue is already dying?
Oh, I see what you're saying. So why would you want to do this diagnostic if you already know that there is the damage is already done?
Right.
So what I'm suggesting is that if you could measure it, so obviously if the ROS is what's driving the microtubules to fall apart, it obviously depends in the process in the cascade where the change in the ROS levels is coming. Is it coming at the end because everything's already falling apart by some other mechanism, and therefore it drives mitochondrial dysfunction, and then at the end you have the ROS? Or is the ROS coming in at the forefront where the ROS is, you get dysfunction in the mitochondria and you're generating more ROS, which is causing the damage, and then everything starts to fall apart afterwards? If it's at the beginning, where ROS is the initial driver of it, as that level starts to go up, then you should be able to pick it up and hopefully measure the emission profile early stages before the damage is starting to be done. So what I'm proposing is that if you can start to notice the incremental change in the ROS levels before the majority of the damage is done, then you would have a early stage diagnostic for these types of illnesses. Okay. Now, again, this is all theoretical, and that's the argument, but that would be the idea.
That I understand. So I actually will get to my second question on this point, but before we go on, I wanted just for me actually to understand the mechanism. So because at the beginning you showed the ROS, the microtubule, and the fact that the protofilaments are sticking together thanks to the tau filaments, right? So the hypothesis here is that as the ROS are increasing, there is some sort of transfer of energy to the tau filaments, which promotes somehow a misfolding or aggregation, and eventually
there's several avenues there. So let's tease them apart. So you have your microtubule, it's stabilized by tau. Now, in tauopathic illnesses, is it that there is dysfunction in tau that then it doesn't stick to microtubules and then the microtubule falls apart? Or is the microtubule being damaged by things like ROS and falling apart, and therefore tau doesn't have anything to attach to? Right? That's, again, these are two open questions. So
I see.
So you kind of see where I'm coming from. This is that we don't know which, it's chicken and egg, which is coming first. And in reality, it's a tied-in system that you can't really, tease it apart. But there's a tie between, your oxidative stress and ROS, the misfolding of tau and it clumping together, and the falling apart of microtubules. And in one way, shape, or form, one can be a proxy measure for the other. So if you have high levels of ROS, which are damaging tau, and tau is damaged, and therefore your microtubules are falling apart, you can then use ROS as a measure for the other two. Or you can say, you measure how much microtubule damage there is, then because of the amount of damage that there is, that you must have some sort of ROS that's going on in the system. Yeah. Does that make sense?
No, completely. And so to close the series of questions, so now, more practical question, right? So we have this ultra weak photon emission, which it's hard to detect in vivo. So how do you measure these UPEs? So my suggestion is, so like, I don't know if there are like approaches or what is the state of the art in terms of actually measuring this kind of radiation in living animals inside the brain. And what if instead we are actually capturing it eventually as a thermal radiation?
Yes, I know what you're saying. So increase, you're measuring like a heat increase in the brain overall. Yeah. And that's a very probable thing. So one of the things that's been argued in this field is that while we're measuring this, you have this ultra weak emission that's coming off of the cell, and it's very, very weak. And it has this tend to shift towards the IR end of the spectra. Is that doesn't necessarily mean that is the amount that's being generated within the cell, right? So you can imagine that if you have this high energy photon and it's trying to get out of the cell, and it starts to interact with things as it's trying to make that progression from the inside to the outside. As it's interacting, it can be losing energy as it goes, and it can be coming down converted from one energy to another. And then finally at the end, it gets emitted. So you may see this low energy light that's coming off, and that is the energy that you're measuring. But the system itself can hold on to that energy and keep it by transferring it around. So like one of the mechanisms I mentioned was tryptophan holding it for these long periods of time in this triplet excited state, or it can be transferred back and forth between a system. There's been work by Del Giudice that says if you have enough chromophores within the system that effectively the photon becomes trapped within the system and can never leave. It just continually gets passed around within the system. So there's a potential for a much greater amount of energy to be localized within the cell, because the photon's always interacting with something that's in there. And that what we are measuring is the stuff that's down converted towards thermal energy, lower energy light, and then that's what we're actually measuring on the outside.
Yeah, thanks.
You're welcome.
We have a question next from Alexandra. Go ahead.
Yes, thank you. Thank you for this amazing talk, Travis. The first question I wanted to clarify something about the microtubules first. So if I understood correctly, the tau misfolding, if there is some quantum process associated with it, it has to be through the microtubules. Is that correct? Because these are the molecules that exhibit quantum processes.
I think I might have confused this point. Is that in your tauopathic illnesses, you have there's something that's going on with the tau. You're getting it's collecting in these clumps. That can either be due to genetic differences in the tau itself. It can be due to ROS affected or interacting with tau itself and causing damage, and therefore making the tau not stick to the microtubule to begin with. Another way that it can form is that if it's produced your body's producing tau and there's nothing for it to stick to, then you have a whole bunch of free tau, and that just might be the natural process where it clumps together because it has nothing else to do. Now, this idea with the photons is it's almost a separate matter. I mean, there could be mechanisms where you know, that energy is being harvested by microtubules and then funneled to tau and damaging it. It could be funneled along tau to some other source, but I'm not sure if that answers your question.
Yes, I mean, I understood the mechanisms with Ross. I wasn't clear where the quantum processes and the energy through the
Yeah, so
emission
that's more looking at how this energy, as it's, you know, being generated by the Ross and it's being absorbed directly by the microtubule, how it could be propagated. Whether that's to cause damage or to serve a functional role, that's up in the air, or even if it's, you know, 100% possible, because again, it's a theoretical description.
And I had another question, more general one. Do you think that quantum processes contribute to cognitive phenomena, and in what way would that be?
Well, I don't know. I'm going to sit on the fence on that one. One thing, okay, so I think that if there are quantum processes going on within a biological system, that light matter interactions is probably the best place to look. I mean, you know, anytime you're dealing with light and interacting with matter, it's a quantum process, and you know, you can get very interesting phenomena that's going on. A lot of the stuff that's been going on with, you know, photosynthesis and light harvesting, again, also with things like the avian magnetic compass. Within the brain, they all involve some sort of photon process in one way, shape, or form. Now, if this mechanism, you know, the transfer of this energy is somehow affecting the way that the neuron functions, so if there is a functional role, then yes, it can play a role in overall cognitive function. So for example, if this energy is being used to control the gating of a channel like the serotonin receptor or something, then yes, you know, it could potentially play a role in cognition. And then I'll end with this. So the one group there, I think it was by Tang and Dai, they had done the work with the glutamate, putting glutamate into the system and showing large amounts of biophotons being emitted. They have another paper that I didn't present here that looks at what they call intelligence levels and comparison to biophoton emission. And so what they did was they looked at, I don't know the animals for sure, but it's something like a worm, a rat, a dog, a monkey, and then a human. And then they look at what the biophoton emission is based on what they would quote unquote an intelligence scale. And they found that, you know, as you go from, you know, worm to mouse to dog to monkey to human, is that you're getting larger emissions of these biophotons. It's an interesting paper. I believe it's in the Proceedings of the National Academy of Sciences, so it's in a high-profile journal. There's a lot of issues with the paper, but you know, there are ideas that you know, the more intelligent or the higher cognitive capacity, the more energy that you're going to be using. So it could be tied in there somewhere, but I'm not gonna rule that 100% at this point, one way or the other.
Thank you. I think there are other people having questions.
Yeah.
I had a few more, but maybe later.
Okay, let me just ask this one question and then come back to you, Alexander, if nobody else speaks up. So there's a question in the chat that I'll read that they asked me to ask on their behalf from Shridi. She says, in my understanding, in your suggested mechanism, the quantum effects are manifested as an incoherent transmission of light. Can there be coherent quantum information processing along with your mechanism?
So it is a coherent process. So the spread of the that initial excitation over the surface of the microtubule, that is a coherent process. So yes, what I'm proposing is that it's initially coherent as as the initial excitation takes place. So when it goes towards I mean, we've done various models of how you know, this light energy could transfer across microtubules. I just showed you a couple of them. One is that one is this natural spread where it spreads across the whole system. We did an additional modeling where the excitation initially hits the system, spreads for the 200 or so picoseconds, and then localizes back on a single tryptophan molecule. But because it is, the tryptophan molecule doesn't de-excite for nanoseconds, then there's the potential for it to then spread across again in a coherent manner again. So it switches between coherent and incoherent. So coherent, this spread, and the incoherent hopping, it, you know, progresses back and forth between the two. And we look to see how far could this excitation travel. And depending on parameters, you know, some of them are, you know, within a given dimer, so it stays very localized. But again, changing parameters within, you know, reasonable limits, you can get it to travel up to, yeah, I think it was 6 to 10 dimers. So again, you're looking at 80 nanometers from where it originally excited. So yeah, I mean, it can switch, I think, between coherent and incoherent hopping.
Okay, thank you, from Sridhi. And I'm going to give the floor back to Alexandra, but what I would say is, like, I mean, you know, if you have to go, of course you got to go, but Travis is willing to stay around a little bit longer if people have burning questions. So I'll sort of officially end it after Alexandra's question, and then anybody who wants to stay, and if Travis has enough energy, yeah. Okay, so go ahead, Alexandra.
Yeah, just a simple one. So I just want to make sure I have this straight. So the aspect that makes the microtubule special in exhibiting this quantum process is that it is the tryptophan. And I was also wondering if there are other molecules in the brain that exhibit similar properties.
Yeah, okay. So one, the reason that it's unique for the microtubules is, yeah, you have the tryptophan, but almost all proteins have tryptophan. It's that tubulin, when it forms a microtubule, it forms it's basically a crystal structure. It's a repeating structure over and over and over again. And you know, there's the density. It's not a very high density of tryptophans by any means. There's other proteins that have, you know, greatly higher densities than tryptophan. But it's that regular spacing and arrangement that continues along the length of the entire microtubule without ending. If you look at actin, it also has a tryptophan molecule, but they're tightly clumped, and then there's a large space, and then tightly clumped in larger space. That doesn't mean that it couldn't support the same sort of phenomena. It's less likely because you have these large gaps in the system.
Yeah, so it's a crystal structure kind of thing.
Yeah, the crystal structure thing is definitely the thing that's unique about the microtubule cytoskeleton. It's regular, self-organizing crystal within the brain.
Thank you.
Okay, are there any other questions on the floor? I'm just taking a look here. I don't see hands up specifically. Okay. Okay, so with that, then maybe I'll officially close and say thank you very much to Travis for a very cool talk. I'm sort of like, I mean, I'm not a physicist by training, but there's a lot of very interesting things in there with quantum, and I look forward to talking more specifics at some later point. So thank you very much, and a virtual clap, or
thank you.
That's the best we could do.
Thanks for having me. I appreciate it.
And yeah, so we're officially closed, and I will shut down the session unless anybody speaks up in the next, say, 5 minutes or 2 minutes, so that Travis could go and get a drink or something. Okay.
Thank you, Travis.
You're welcome.
Okay. Yep. All right. Yeah. Okay. Yeah, actually, one thing I could just sort of like just ask a bit more about this optogenetics, right? I mean, like when it came out, like, I mean, I remember being very excited, like just from a modeling perspective, because, oh, we could target particular cell types and try to see how they contribute to the circuit and all the rest of it. And then I forget exactly, but there have been subsequent studies showing, you know, that you have to kind of take it with a grain of salt, you know, because people are trying to draw causal effects with the the optogenetic studies, and so proper controls weren't being done, it would seem, in some of the experiments. And now from what you're saying, I think, okay, of course there's different wavelengths that are used, right? And depending on if they want to differentiate different cell types. So now I'm kind of even more worried, so to speak, in terms of how good it is. So I don't know if you interface or interact, like given
no, I haven't. I mean, that one paper, that one that just came out last year with the violet light, that's effective. I mean, that itself is like an optogenetic experiment, but they're using a natural light sensitive protein.
Okay.
Right. So that one I think is a little bit closer to something that could actually happen.
Okay.
I mean, I think it just baffles me why you would have a light sensitive molecule deep within the brain.
So obviously you think it's not waste.
I don't know. I like to err on the side that it evolved for a reason. It might be just artifact, but let's not rule that out, or let's not jump there to begin with.
Yeah, and actually, just to follow up on that, like, I mean, in terms of the studies, like, I mean, these molecular dynamics and such studies are, I guess, huge. I haven't done them myself, but I mean, part of it is just a huge computational exercise effort. So, and depending on if you include what and stuff, and this is speaking from a sort of naive point of view, is it practical to really think about being able to do those kind of, like, well, I mean, the computing is increasing all the time, like, availability.
Yeah, it depends on which scale you're talking about. Like, I mean, I know that they can do fairly large proteins. Are getting towards quaternary structures, so bigger proteins. But there's depending on how much detail you want, there's always the trade-off between detail and size of the system.
But
right?
Don't you need a lot of detail for your kind of simulation?
Oh, so I mean, it depends on, like, we do a lot of coarse grading. So for example, when we were calculating out the excitation energies of the tryptophans, right, we're using density functional theory, which is like a quantum chemistry calculation, which is really computationally intensive. But so we couldn't do it for the whole. Ideally, you would want to do it for the whole tubulin protein all at once, but you can't. Or at the time we couldn't. I don't know if you can do it now. It's getting there. So we look at just the tryptophans and then take those values and then apply it to the next level up, which would be like a molecular dynamics type model. Now, if you wanted to go from a tubulin protein and do molecular dynamics up to a whole microtubule, you have to then start coarse grading it, and you wouldn't be able to look at each individual atom at that point because it's just way too much to calculate. Right, so you can assume that the surrounding protein is some sort of continuum, and that you only would run then just the tryptophan molecules, right? There's trade-offs wherever you do it.
So does it make sense to think about, and again, I'm coming from a point of naivety, if you were looking at different drugs and stuff. So I mean, like, you probably, in talking to Don, sort of familiar with the sort of studies they do, right? So if you're trying to see if this biophoton type change or whatever, this quantum effect is important for different drugs, does that make sense to even be able to explore?
Most of my work is trying to come up with drugs and mechanisms to prevent the kind of ROS related damage that we're seeing.
Okay.
So a lot of it is looking for inhibitors and or designing drug treatment courses to stabilize microtubules while reducing inflammation at the same time, that type of stuff. This thing here, the whole thing with the light traveling along the length of the microtubule, that's kind of like I said, it's like a blue sky, just something that's interesting that we're looking at. It. How it would tie directly into clinical usage is kind of still tenuous.
Okay.
Right, but in some one way, shape, or form, it's basic research as opposed to applied. Yeah.
Okay, well, we need that. Okay, thanks. I see Idir there, like, you want to ask a question?
Hi everyone. Thanks, Travis, for the presentation. This is the first time I discovered this technique. So I have two questions. First one, it's related to the hardware accelerator. So you mentioned that the simulation needs lots amount of computation.
Okay, I just see this.
Yeah. So I was looking around. I found just one paper. It's master thesis here at UOFT. So I'm just wondering, have you ever used something like accelerator?
No, I haven't. No,
No,
I'm looking at this right now.
Okay, what are you using? Are you using a console, FM, or finite limit method for simulation?
For simulation, so I'm using the supercomputer down at the University of Miami.
Okay.
Right. So I'm using so the software packages that I'm using for the molecular dynamics simulations or the quantum chem calculations, they're software packages that have been optimized to be used across. They're parallelized to be used on the supercomputer. So it's a parallel application, and we run it. As far as coding our own simulations for photon interactions, this is new to me what I'm looking at here. So thank you for sending this. This is quite.
No, this is not pleased. My second question is, can we consider this like a thermal therapy, or is it because during my PhD was thermal ablation of tumor. So I investigated different techniques. Then I applied for RF ablation. And I used COMSOL for the simulation of the ablation, blah, blah. Then the bio-heat transfer equation, Penn's equation. Then when I started reading here, I found that you have a reflective equation, blah.
Yeah.
So can we use this for ablation of tumors? Is it like a technique used currently? I'm just a little bit confused because you mentioned several times damage.
Yeah.
And
yeah, so you're talking about to get rid of tumors. I don't think so. The damage is okay. So for like a tumor, you would want to cause significant damage in a short period of time. And I think the type of damage I'm talking about is a slow, progressive buildup of damage over a long period of time. Yeah.
So I mean, for ablation,
if you want to think about it this way, the type of damage I'm talking about is the same as radiation type therapy.
Yes.
Right. It's just very, very, very weak.
So for the ablation, you're right. It should be short and high. But for hyperthermia, the temperature, it should exceed like 43 to 45 degrees Celsius. Okay. But for longer duration. So we expose the tissue, the targeted tissue, for for hour, for 45 degrees, or then the destruction process comes slowly. But so I don't. Just by reading all this, I was googling all the techniques. Then it comes to my mind. I don't know if it's good.
Yeah, no, I'm not sure. I know in regards to cancer. I mean, I've seen some papers where they're using biophotons, but again, I think it's more for detection, for detection of the cancer, not for treatment. I'm not looking for some sort of pathway that you can exploit to halt tumor progression.
Okay, thank you so much.
So this is just right off the top of my head. I don't know much about cancer. I'm not much of a cancer researcher, but as these tumors grow, do they not? I mean, there's got to be inflammation and stuff. How do tumors protect themselves from inflammation and ROS damage and all that?
I really don't know.
Right. Yeah. Okay. All right. Just a question there. If you have an inflamed area, then why are they not?
Yeah. The thermotherapy that I have been worked in, it's like targeting area. Then there is a science developed called dosimetry. So we have to define the right dose, the frequency, the amplitude, and the right location for the probe, or if you want, use microwave or laser or something.
Right,
this is like this is what we use, but for this photonic therapy, this is first time I discovered.
Okay, excellent. Thanks for the.
Thank you. Thanks, Francis.
You're welcome.
Hi again. Thanks, Travis, for the great talk. And my question is, do you think if you implement QM and MM, you will get something, I mean, more precision, or it's not useful?
I think you get something more realistic. I mean, you know, okay, so for example, so grossly, the kind of stuff that we were doing, the way that we did it, you know, it says that there's a potential for it depending on the way that the things are parameterized. If you were to add in more detail, you would just get, you know, finer understanding of actually what's going on. So
because for dipole-dipole interaction of this tryptophan, I was wondering, I don't know, this geometry of this microstructure probably will affect the things, and probably doing side molecular dynamics would help.
yeah, I mean, yeah. So again, like, so the stuff that we did it on a snapshot. So it was a single still frame. So we did the molecular dynamics simulation to get the
optimized configuration,
optimized structure, right? So to be representative of what it is in biology, like in the biological system, as opposed to a crystal structure. But then if you were to do a you know, full-scale molecular dynamics simulation with the tryptophan excitation, you could get a better understanding of how that excitation could propagate along the surface of the microtubule, or could you know, hop between the tryptophans. I know there's work by Patrick Callis or Calais, I'm not exactly sure to pronounce it, where he did just that, where they did tryptophan excitation in, I forget which type of molecule, but it was coupled with molecular dynamics. So what they would do is they would simulate for a period of time, and then they would take a snapshot of the molecular dynamic simulation, run the quantum calculation on it to see where it would want to go at that point in time, change the parameters, put it back into the molecular dynamics simulation, and then continue on to another period. And then they kind of intersperse between quantum dynamics and molecular dynamics back and forth to try and get a better description of what happens with the tryptophan excitation. I mean, that's something that could be done with tubulin itself or any other protein.
And one last question: this talk is recorded, right? Because there are a bunch of people in Calgary. They are sleeping, so they were asking me, is it possible to have this talk?
Yeah, I did record it, Hadi. Oh, let me put my video on here. Actually, I don't know you, Hadi. I guess you're from Calgary?
Right.
Oh, okay. All right. Yes, I did record the talk with Travis's permission.
Yeah, do you work with Christoph Simon?
Right, Race.
Yeah. I've spoken with him a few times. His work is great.
Yeah, we were just told that there are some experts on that. We were motivated, and this was really good talk. I learned a lot. Thank you.
Okay, so with that, yeah, so thanks a lot, Travis. So I did record it, and with your permission. And so what I'll probably do is, you know, like on our KCN Hub where I advertise the KCN event, I'll just add the link there.
Okay.
For your talk. So you're just to confirm you're fine with the.
Yeah, no, I'm fine. Yeah, sure.
Okay. All right.
Okay.
Okay. So thank you very much again. And even though this is not ideal in terms of interaction, I think it would, you know, we were able to get a lot of interaction still. So. Well, it was the topic and the speaker too, of course. So, so thank you.
All right,
and,
thanks very much.
Talk again soon.
We'll talk again soon. Thanks a lot, Francis. Bye-bye.
Take care. Stay safe.
You too.